Imagine stripping the membrane off a living cell. Water, ions, nutrients, and proteins instantly scatter. The cell immediately loses control of its internal environment, and homeostasis collapses within seconds. The plasma membrane prevents this. It acts as a highly selective, dynamic boundary that constantly dictates what enters and leaves the cell. The foundation of this boundary is the phospholipid. Notice the structure. It features a hydrophilic or water-attracting head attached to two hydrophobic, water-repelling fatty acid tails. Because water exists both inside and outside the cell, these molecules naturally self-assemble. The water-repelling tails tuck inward to hide, while the water-attracting heads face outward. This specific orientation creates a stable, double-layered barrier, the phospholipid bilayer. Biologists describe this structure using the fluid Mosaic model. It is a mosaic because various protein components are embedded throughout the lipid layer, and it is fluid because the phospholipids and proteins constantly slide past one another laterally. This chemical architecture creates a flexible, shifting barrier rather than a rigid, impenetrable brick wall. The internal hydrophobic core dictates what can cross. Small, nonpolar molecules, like oxygen, pass through the lipid bilayer directly and effortlessly. But watch what happens to charged particles or large polar molecules. The hydrophobic tails repel them entirely. For these rejected molecules to enter or exit, they must use specialized transport proteins that act as specific doorways through the membrane. Ultimately, a molecule's physical properties, specifically its size and charge, determine exactly how it moves across the boundary. When particles move down their concentration gradient, flowing from high concentration to low, they use passive transport. This process costs the cell absolutely zero ATP. If a substance slides directly through the lipids, it is simple diffusion. If it requires a transport protein to provide a clear path, it is facilitated diffusion. When water diffuses, it is osmosis. Water naturally flows toward the greater solute concentration. We use tonicity to predict volume changes. In a hypertonic solution, water rushes out, shrinking an animal cell. In a hypotonic solution, water rushes in, swelling a plant cell. In an isotonic state, solute concentrations are equal. The molecules do not stop moving. They simply cross the membrane in both directions at equal rates, resulting in zero net change. Passive transport relies entirely on these natural physical gradients. The cell maintains this movement without spending any of its own energy. Sometimes a cell must force substances against their natural concentration gradient, moving them from low concentration to high. This is active transport. The flashing protein pump requires direct cellular energy, usually ATP, to push ions upward. Cells also use cotransport. As one molecule falls down its gradient, the pump harnesses that momentum to force another molecule up its gradient. For massive payloads that cannot fit through protein channels, the membrane folds inward to engulf large external particles in a vesicle, a process called endocytosis. Conversely, the process of spitting material out of the cell is known as exocytosis. The membrane relies on two final components to function. Cholesterol regulates fluidity across extreme temperatures, and surface carbohydrates act as cellular recognition tags. To control its internal environment, a cell must actively spend its energy reserves to defy natural equilibrium. AP reasoning depends on applying these rules to experimental evidence. If a scientist blocks ATP braction, but a specific molecule continues entering the cell, we can infer the mechanism is passive facilitated diffusion. If transport halts completely the moment ATP stops, we deduce the process relies directly on active transport. Consider a mutation. Changing a transport protein's amino acid sequence alters its physical shape. Because protein structure dictates function, that altered shape destroys its ability to transport its target molecule. This cause and effect chain defines the logic of the system. Identify the gradient, state the direction of movement, name the mechanism, and justify the energy requirement based on the data. In practice, a cell in a hypertonic solution loses water. Protein pumps indicate active transport, and while equilibrium stops net change, the molecules themselves never stop moving. Permeability follows a consistent logic. Small, non-polar molecules cross the bilayer directly, while anything large or charged requires a dedicated protein channel. By mastering the mechanics of the fluid Mosaic model, you gain the ability to accurately predict molecular behavior and transport across all biological systems.